If we look into the diagram of this cell cycle, we can easily see the cyclical changes, the sequential changes that occur in the cell during a cell cycle. So initially these are the cells. This is first cell, this is the second cell. So let's consider the first cell. So initially it was like this. Then when it enters into the G1 phase, in G1 phase growth of the cell occurs. So the cell increases in size, and also some other factors are monitored, as I have already mentioned what factors are monitored. Then comes the S phase. After the completion of G1 phase, the cell will enter the S phase. So in S phase, what happens? DNA replication will occur. The genetic material of the cell will be duplicated in S phase. And after the completion of duplication, that is after the completion of S phase, the cell will now enter the G2 phase. What happens in G2 phase? The cell will monitor whether the DNA has replicated correctly or not, whether the factors in the environment are favorable for cell division or not. So these are some conditions that are monitored in the G2 phase. Now if the conditions are accurate, if everything is all right, then the cell will enter the mitosis phase, which is the cell division phase. So mitosis is composed of two phases. First phase is karyokinesis or nuclear division, and the second phase is cytokinesis or cytoplasmic division. After the completion of these two processes, we can conclude that mitosis has completed, and we can finally get two daughter cells from a single parent cell. So this is the entire cell cycle. What are the roles of cyclins and CDKs? So when the cell is going through the G1 phase, CDKs or protein kinases required for the progression of the cell into the S phase will be synthesized. So their levels will go up. And once the cell enters the S phase, the work of this cyclin CDK complex, the work of this CDK, these protein kinases action are over. So these proteins must be degraded. Once their work is over, they have performed their function, so they will be degraded by the cell. So there are different mechanisms by which these protein kinases can be degraded. So the basic concept is that after the completion of function, the proteins will be degraded. Now the cell enters the S phase, and what type of CDKs are required in this phase? So the name of the CDKs are SCDK. So in this phase, in S phase, when the cell is going through the S phase, these SCDKs will be synthesized. Their levels will go up, and after the completion of all the events of the S phase, the cell will progress through the G2 phase, and the function of this SCDK is over. So now this CDK will be degraded. Again, when the cell crosses the G2 phase, when the cell has performed all the necessary events that are in the G2 phase, all the events in the G2 phase are completed. So CDKs responsible for G2 phase will also be degraded. And what will be synthesized? The M phase CDK. The CDK responsible for progression into the M phase. CDKs that have function in the M phase of the cell cycle will be synthesized. So in late G2 means at the end of G2, the levels of M CDK will increase because they have function in M phase. And after the function is over, after the completion of M phase, these M CDKs will also be degraded. So sequential changes. What happens? At a single time, the level of a particular CDK will be higher, and no other CDKs will be present in that amount. In a cell, only at a time, a particular CDK will be present at a higher amount, and after their function is over, they will be degraded. So this is the concept of cell cycle. In a single round of cell cycle, only a single round of DNA replication must occur. Right. So in the diagram, we can see that only in S phase, initially what was there? Initially the cell contained a single number of DNA. For simplicity, I have denoted here only a single DNA. Suppose this is a single DNA. A cell contains only one DNA. So in the G1 phase, the cell will contain one DNA. In S phase, duplication of the DNA will occur. So one DNA will be duplicated, and now the total number of DNA is 2. In G2 phase, there is no change in number of the DNA. So in this G2 phase, the cell contains 2 DNA. After the completion of M phase, what happens? Cell division happens. After cell division, these daughter cells will contain a single DNA. So this is the concept. Initially there was one DNA. So if number of chromosomes is denoted by N, number of chromosomes is denoted by N. So in G1 phase, number of chromosomes is N. In S phase, number will be 2N. In G2 phase, number will again be 2N. So G2 phase will also have 2N number of chromosomes, and after the completion of M phase, individual daughter cells will have N number of chromosomes. So both the cells will have N number of chromosomes. So this is how the number of chromosomes is maintained in the cell cycle. And from this diagram we can easily see that in a single cell cycle, one round of DNA replication occurs. And this is a very crucial event, and this event is very tightly regulated. So we will see the regulation mechanism of this process.